talk about fungology, and this is going to be the introduction. First thing, what is fungology, also known as mycology? It's a study of fungi or fungi. And fungi includes molds, yeast, and mushrooms. We need to know fungi are eukaryotic cells. If we say they are eukaryotic cells, we mean they are similar to humans. They have got membrane bound organelles, for example, the nucleus and the mitochondria. They also have ribosomes, but the ribosome, of course, even prokaryotic cells have got ribosomes. But in prokaryotic cells, we have got the 70S ribosome. In fungi, there is the same ribosome that is found in human, which is the 80S. That is why it is difficult to treat fungal infections, because nearly the structure for fungi is nearly the same in all aspects with that of the human. So drugs that work on fungi are also going to work on human cells. But a little difference that we're going to utilize. Fungi have got a cell wall that is made up of carbon, hydrogen, and oxygen with a chitin, glucan, and mannan. So we are going to take advantage of this because humans don't have this glucan and the chitin in the cell wall, right? And another thing we need to know is that the cell membrane of fungi has a ghosterol. We also take advantage of this because in our cell membranes we have got cholesterol. And so you are going to see some drugs like the imidazoles that will work on this ergosterol. We'll talk about them shortly. You also have what we call the polyene and fungos that will bind to ergosterol. And so that is how these drugs will be able to work. Another thing we need to know about fungi is that they are heterotrophic, meaning that they require organic carbon for them to be able to live, right? Again, they are also saprophytic or saprobic. They live on dead material. Besides that, some of them are parasitic. What does that mean? They're going to live on other living organisms, not on what is dead, like saprophytic, but on what is living. Now that we have done some bit of that, another thing we need to know is that fungi are dimorphic. They exist both in the mold form and yeast. Okay, you can see this is the mold form. These forms that you can see, these are the mold forms. And then you have got the yeast. This is the appearance of the yeast. Yeast is the form that you find in the body in vivo. You can see that is at body temperature. While the mold is the form that you find in the environment. Nearly all the fungi, they are going to be dimorphic. So now that we have talked about a bit of a structure, if you look again at the structure of fungi, let's get it microscopically. We have got the cell wall, and we also have the cell membrane. There is our cell membrane. Let me put them here, this side. So this is the cell membrane. So what's the importance of us understanding this structure in regard to the antifungals? We said that the cell membrane is made up of what? Ergosterol. Humans have got cholesterol. These ones have got ergosterol, which is a sterol. Now, how do you make this ergosterol? So for you to make ergosterol, you start with what we call squalene. This squalene is going to be converted into what we call lanosterol, and then the lanosterol is going to be converted into ergosterol. We have got enzymes that do this work. So for the squalene to be converted into lanosterol, we need what we call squalene epoxidase. Epoxidase. And for us to convert lanosterol into ergosterol, we need the cytochrome 1 alpha demethylase. So that is CYP450. You know, in our liver, we also have this cytochrome enzyme. So this is the 14 alpha, not 1 alpha, but 14 alpha demethylase. So you are now going, it is on these that you are going to have some drugs that are working there. So the first, let me start with the first class of drugs, which we call the polyenes. Okay, number one class of drugs are polyene antifungals. How do polyenes work? How polyenes are going to work is that they are going to come and bind to a sterol. They bind there, and once they bind to a sterol, they will now form pores. And you know, when you form pores, what happens that now fluids can enter the cell, and then it can destroy those fluids that are entering the cell, can now destroy it. So what are the examples of these polyene antifungals? So we have got amphotericin B, amphotericin B. We also have what we call nystatin. Just something for us to know about these drugs. For the amphotericin B, this one you mostly use it for the treatment of cryptococcal meningitis. You're actually going to combine it with another drug which we call flucytosine for candida infection and cryptococcal meningitis. And you'll mostly give it IV because meningitis, you need the drug to reach the cerebrospinal fluid, or to reach the brain, not cerebrospinal fluid. That's why I want to give it IV, right? And this drug has got a certain side effect, which is known as a shake and bake side effect. And when you give this drug, it actually gets to, when you give a lot of it, it has got effect on renal function. That's why when you give amphotericin B, you want first to give what we call a test dose to be able to tell now the amount of amphotericin B that you need to give. Okay. But besides that, the shake and bake that I've talked about, this as a characteristic of fever, chills, rigor, you know, if someone is dead, the way the muscles contract, and hypotension. So you can give meperidine to prevent that rigor. The side effect for amphotericin B, I've said, is nephrotoxicity. Okay, and how can you reduce the nephrotoxicity? You can give a loading dose with normal saline. You can as well combine with another medication so that you reduce the dose of amphotericin B, and you can also use what we call the liposomal amphotericin B formulation. But this form requires a high dose, though it has got less toxicity and is expensive. Then statin, the other drug, which is a polyene, this one is used to treat topical. It's topical, like you apply it on an area for yeast infection and also skin and mucocutaneous infections. You also have some other drugs. Now, other drugs that you are going to give in fungal infections, you are going to give those that are going to come and prevent the conversion of squalene into lanosterol. So those drugs are working here. What do we call those drugs? We call them the amines. Amines. So these ones we are going to see the end in FIN. For example, nafrifine. Another one, butenafine. Another one, amorphine, tebifine, just like that. And these ones, you mostly use them for dermatophilic infections. We'll talk about these infections. Dermatophilic, these are infections of the skin. Dermatophilic infections like onychomycosis. Okay, so those are the ones of the toenail and fingernail. Now, we also have those drugs that are going to inhibit the conversion of lanosterol into a sterol. So these ones are working here. So these drugs, we call them the azoles. Now, because azoles inhibit cytochrome P450, azoles will not only inhibit this cytochrome P450 of the fungi, instead they also inhibit the cytochrome P450 in our liver. That is why these antifungals that work on the azoles, for example, ketoconazole, we call them liver enzyme inhibitors. So if you give a drug that is metabolized in the liver, you also give these antifungals that inhibit the conversion of lanosterol to ergosterol. It means that they inhibit a liver enzyme, okay, and it means that that other drug that you've given that is supposed to be broken down, metabolized in the liver, will not be metabolized. What does that mean? It will increase the toxicity of that drug because it will still be in the body. All right. Hope we get that. Now, for these azoles, we can divide them into two many groups. We have got what we call the imidazoles. Imidazoles contains two nitrogen atoms. We also have what we call the triazoles. Even from the name, the triazoles contain three nitrogen atoms. So what are the examples of the imidazoles? Imidazoles, we have got ketoconazole, we have got myconazole, we have got clotrimazole, okay, and then we also have B4-comazole, KMCB. And then for the triazoles here we have got things like itraconazole, we have got posaconazole, we have got fluconazole, we have got voriconazole. And voriconazole is a systemic drug, so you give them for systemic infections. It's the drug of choice for systemic infections. So please, you can just know the imidazoles. These are three: ketoconazole, miconazole, and then clotrimazole and bifoconazole, KMCB or BCKM, and then the rest will be the others. Okay, hope that makes sense. So besides these drugs which are here, which are working on the cell membrane, you are also going to have other drugs that work on the cell wall. Remember we said that the cell wall for these fungi contains what? Contains glucan. So we'll take advantage of that. So we have got the drugs which we call echinocandins. And how do these echinocandins work? These echinocandins, they are going to target the beta 1,3 glucan synthase that is found in the cell wall. And this one is going to be the enzyme beta 1,3 glucan synthase. This is the enzyme that makes beta glucan that is found in the cell wall. And so once they inhibit this enzyme, they are going to stop the synthesis of the cell, and that's how they are used in the treatment of these infections. What are the examples of the chinocandins? These ends in fungin. You have got caspofungin. You have got also micafungin. You also have what we call anidula fungin. Okay, so these are the drugs that are going to inhibit the synthesis of beta glucan. These are used in the treatment of invasive fungal infections. Okay, the treatment of invasive fungal infections together with amphotericin B and fluconazole, especially in conditions where there is amphotericin B and fluconazole resistance. That's when you give these echinocandins. And these echinocandins, some of them are fungistatic, they kill bacteria. Others are some of them just prevent the growth of bacteria. That's fungistatic. Others are fungicidal. For example, they are going to be fungicidal to Candida, but they are going to be fungistatic to Aspergillus species. Okay. You also have what we call antimetabolites. Antimetabolites, these are antifungals that will just prevent the formation of these. They will be working on DNA synthesis and the RNA synthesis, those steps. So we have got some drugs here. We have got glycofulvin, Glycofulvin disrupts the fungal microtubule, so it disturbs cell division. That is how it works. And it is only active against the dermatophytes. It's also hepatotoxic. You also have what we call flu cytosine. Flu cytosine is first going to be deaminated or converted to 5-fluorouracil inside the fungal cell wall, and this one inhibits nucleic acid synthesis, and that leads to cell death. This drug is well absorbed and is active only against the yeast form of the fungi, and it is less toxic in comparison to amphotericin B. So this is a brief on the antifungals. So another, continuing with the fungi, another thing we need to know about these guys is that they have got their own kingdom, which we call the fungi, and they are able to be multicellular. They can have a lot of cells, okay, and they'll comprise of some of them. They have got filamentous appearance. You are going to see them like that. We call that as a mode. Others are unicellular in nature. They will just appear like that. These are the yeast, and they are the ones that you see in the human body. The mode is going to be in the environment. Some are dimorphic. Actually, majority of fungi are dimorphic depending on the environment. Okay, almost always yeast is in infection because that's what is found in the human, and these fungi can cause superficial infections, cutaneous infections, subcutaneous infections, and also systemic infections, and are typically free living. They are acquired from the environment and are part of the normal flora. They cause disease by inflammatory response. So once you have got fungi in the body is responding to the fungi by causing inflammation. That is how disease happens. Besides inflammatory response of the body, also by direct invasion, they'll simply penetrate uh, epithelial barriers and also destroy tissue. Again, fungal infections, we call those mycoses. They are classified depending on which part of the body they affect and the type of infection they induce. For example, I've got endemic mycosis, okay, from geographically restricted, and these are true pathogens. So the endemic mycosis, these are true pathogens. They are found, they always cause infections. And then opportunistic mycosis from non-true pathogens, these ones are non-true pathogens. They cause severe disease only in the immunocompromised. Those are opportunistic. Subcutaneous mycosis, you are classifying them now with the type of infection that they induce. So subcutaneous, they cause infection that is just under the skin. Okay. So disease involving the skin, subcutaneous tissue, and lymphatics. You also have the superficial and cutaneous mycosis. These are common, limited to the skin and the skin structures like the hair. These are superficial. Fungi belong to the fungi kingdom, also known as the Eumycota, and are classified on the basis of the mode of sexual and asexual reproduction, morphology, the life cycle, and the physiology to some extent, how they really work. And these comprise of at least over 100,000 species, and they are found worldwide. Most are ubiquitous in nature, and less than 1% cause disease in man. Most of them do not. They play a crucial role in biodegradation of waste material. For example, if something dies, fungi grows there and starts to break it down. So that's biodegradation. They are also economically destructive. For example, you are going to find them on bread. That's not nice. Okay, because you are going to buy another bread, so economically destructive. They cause widespread damage to food and fabrics, and they are used also commercially in industrial processes such as fermentation and in the production of important metabolites such as antibiotics and steroid hormone derivatives. So these guys are eukaryotics. They have got a nucleus, cell membrane with a lipid, glycoprotein, steroids, mitochondria, and then they have got also ribosomes which are bound to endoplasmic reticulum. They have got a cytoskeleton, microtubules, basically the same way the human cell is. They have got cell walls with chitin, mannan, and glucan. In nature, they derive nutrients from decaying matter. They don't have chlorophyll, that's why they are not plants. Okay, and so they don't undergo photosynthesis. They are almost all strict aerobes. They love oxygen. Strict, nearly all of them. And favor 37, 35 to 37 degrees Celsius of temperature. That's the temperature at which they are mostly going to survive. They exist in two forms: the filamentous form, that's the mode, and that's the multicellular organism that's found in the environment. And then the unicellular is the yeast that's found in the body. The modes, these guys branch. Okay, they go like that. They have got branches. They have got, those branches we call them hyphae, and then collectively that's known as the mycelium. Hyphae may be septate, they can form septa, that is, those are known as partitioned, or they can be co-enocytic, multinucleate with no cross walls. Yeast are single cells, ovoid or spherical with rigid cell. So the yeast appear like this. Single cells, ovoid in appearance with a rigid cell wall. And the yeast will divide by budding or binary fission. So you start with one and then it will end up dividing into two. That's binary fission, that's asexual reproduction. And some will produce polysaccharide capsule. You will see the importance of this capsule when we start discussing individual yeast. So I mean individual fungi. The yeast form, there's multiple solitary small round, these are multiple solitary small rounded forms actively dividing, growing, and metabolizing form. So this is the form that divides. Reproduction occurs by budding or binary fission, and the colonies are typically moist and mucoid. You can see how they look, actively dividing. Okay, so this is the budding yeast, and then this is the saprobic phase or saprophytic phase. This is the yeast that is not dividing. This is the one that is going to be used for feeding. When there's something dead, that is the one that you are going to find in dead organisms. Okay. So yeasts often show multiple buds. You can see you have got one and then they have got things that are coming out like that. Those are the buds. And then the yeast capsule is extremely large. You can see the capsule there, very large. On agar, yeast forms moist colonies that appear to but are larger than bacteria. So they appear like bacteria on these colonies. Okay, when you are growing the yeast, but they are larger. They grow as microscopic branching thread-like filaments. Okay, when you talk about these fungi, in this case, these ones are the mods, which have got filaments, and the filaments are known as hyphae. Hyphae collectively will be called the mycelium. The hyphae grow by elongation at their tips. So if you have got something that is just ending here, so you to start from here growing, okay, are the tips. At the end of hyphae are rounded forms that could be confused with yeast, which are called the conidia or spores. So at the end of these hyphae, at the end, you are going to see the conidia, also known as the spores. So this is not a yeast, so you shouldn't be confused. Hyphae are either septate or conocytic. Septate, they form septa, And many display both forms and are able to exist as modern yeast. They are known as dimorphic, okay, and occur when fungi infect living host. The yeast is a parasitic form. That's the one that is found in the body. And occasionally this rule may be reversed. Sometimes the mode can also be parasitic, but majority of the times it's a yeast that is parasitic. You can see this is the mode, this is the yeast. The yeast, they are like that, just round oval. The mode, these are the ones that have got hyphae. So how do humans encounter these fungi? So it is by two ways. You have got those which are freely living in nature or in the environment, and they are going to enter into our environment. So it is just by two means. It's either the fungi enters our environment or we invade their environment. That's how we are going to get the infection. So those which are free living in nature, they can enter into, maybe we can enter into their geographic environment, confinement, and then naturally we can encounter them when we are immunocompromised. Those are free living in the environment. Okay. So some of them, they are part of the environment and we're supposed to survive with them. But once we become immunocompromised, they start causing problems. Or maybe they are properly in their environment and we are not used to them, and then we end up entering into their environment. Once they enter into us, it becomes a problem. And majority of them are going to be found as part of the normal flora of our body. And so they have got the ability to cause severe infection in immunocompromised individuals. Because they are part of normal flora, they don't cause infection. But once we become immunocompromised, that's when the problems begin. How do they enter the body? Innate immunity protects us from fungal infection. You know, innate immunity is the first defense. For example, this is the GIT. The GIT contains a lot of things. There is a epithelial barrier that prevents invasiveness, entry into our cells. Okay, you also have the acid that will be protecting us, so that's innate immunity. The skin, the mucosa surface, these guys protect us. That's, they limit the infection. Desiccation, epithelial cell turnover, fatty acid, low pH bacterial flora, plus mucous membranes, these help in getting to keep the infection self-limiting. Okay. If there's any alteration in normal flora or nutritional change, for example, you have got these Candida albicans, once there is a change in nutrition, they are going to be able to proliferate and may now allow the entry into and into the body that leads to infections. Violation of natural barriers, for example, when there is trauma, foreign bodies, this will allow the entry of these fungi into the sterile area of the body. The sterile area, this is the area that you don't find microorganisms, for example, in the blood. And once that happens, infection happens. How do they spread and multiply? So tissue reaction to fungi, to fungal presence varies. For example, with a species number one, so some species are more going to cause a worse infection than others, and also the duration of infection. If the infection has been in the body for long, you'll see that the tissue reaction becomes worse. Okay, so the species and the duration of the infection. Some mycoses are typical of causing low-grade inflammatory response that fails to clear the fungi, leaving a low virulent infection. Okay, that is just the species of the fungi itself. It will just cause a low grade inflammatory response because of the species of the fungi itself, and so it will not be all cleared up when there is this low grade inflammation, and so there is low virulent infection. Okay, some of it will actually be cleared because there's a low grade inflammation, but then some will remain causing this low virulent infection. It's not too bad. And then there's also a generalized non-specific inflammatory reaction, which is very critical in eliminating the fungi that can happen. Neutrophils, you know, neutrophils are involved in acute inflammation. These are phagocytic cells, and they are also going to be used in killing of the fungi. that enters the body, and that is the most important mechanism by which these fungal infections are going to be managed by our natural body or by our cells. Phagocytosis may fail with some fungi, especially those which have got a capsule, because the capsule prevent them from phagocytosis. The fungal cells are highly antigenic. An antigen is a protein that will cause our body to produce antibodies against it. So since these are highly antigenic, it means that they are going to provoke our immune system, both a cellular and humoral response. The cellular response is by the T cells, the humoral is by the B cells. B cells will produce antibodies. The T cells will go and now attack the same antigen. So antibodies and the complement will play a role in extracellular killing of some fungi, and these ones will secrete the lysosomal enzymes, and lysosomes are going to be involved in digesting the fungi. Resistance to fungal infections is due to cellular or T lymphocyte mediated immunity. So sometimes we can be resistant to not get sick, to not have these fungal infections. Why? Because of the T cells and also the B cells. So that's the cellular and humoral immunity that will help. So how do these fungal infections damage our body? So the outcome of the infection by fungal infections is determined by number one, virulence of the infecting organism. How bad is it? And then the size of inoculum. How much of the fungi do you have? And also adequacy of host defense. If someone is sick, it means that their defense is not such strong, so it will worsen the condition. Tissue damage varies with each species and the organ that is involved, but typically is due to direct invasion of organism plus associated displacement or destruction of vital structures and toxic effect of inflammation. So what is leading to this tissue damage varying in different individuals is number one, because of the effect of inflammatory response, the toxic effect, number two, the destruction of these vital organs, okay, and also the direct invasion of the organism. These are and also the species, okay, and the organ that is involved. Because if organ one is involved and not organ two, sometimes infection might not be that bad. The fungi that cause invasive disease are not known to secrete toxins. Okay, sorry. Fungi that cause invasive disease are not known to secrete toxins harmful to host. Invasive disease means this is the epithelium. Here is the fungi. It can penetrate the epithelial barrier to go into the body. So such fungi that penetrate epithelial barriers, they don't secrete toxins. Okay, they are not known to secrete toxins. How do you diagnose fungal infections? Number one, you can use labs. For example, microscopy. Some are large enough for direct examination. You can just look at them microscopically without using a microscope. Okay. You can also look at the tissue, that's histopathology. You can culture some of them. You can also look at blood serology. You can also detect the antigen of the fungi that you are dealing with. Morphological characteristics can also be distinctive enough in some to be able to aid in identification. So you just see something that is like that. Obviously, you are guessing this is fungi. But this is not a form that is found in human, because the form found in human is the yeast. So the morphology can help in some systemic infections to provide immediate diagnosis. General culture grow of fungi from sterile areas implies infections. Culture must guide, okay, and differentiate from contamination. So if you culture the bacteria, the fungi, it grows, you should be able to differentiate it from contamination. Maybe the culture itself is just contaminated by the bacteria, by the fungi, and it's not what came from the body that you are growing. So you need to differentiate them. Serum antibody detection is also useful in diagnosis of endemic infection. The endemic ones, these are the pathogenic infections, but it's rare. You don't use antibodies to diagnose opportunistic infections, because normally you don't have antibodies produced against these opportunistic infections. And direct detection of fungal antigens proving more useful. Just the antigens themselves. Polymerase chain reaction is also useful in getting to diagnose these infections. How do you treat mycotic infections, fungal infections? Not all of them require treatment. For those that require antifungal agents, toxicity is a problem because of structure and pathway similar in fungi and mammalian cells. So if you give this drug, instead of it only working on fungi, it's also working on our cells. So thus there are very few antifungal agents which are available. Okay, in conclusion, fungi comprises of over a hundreds, hundreds of thousands of species. They are eukaryotic, and these guys, many encounters with fungi do not produce disease. Okay, however, large infection dose and immunocompromised conditions are enough to produce disease, actually serious and life-threatening disease. A variety of diagnostic tests are now available to be able to determine the presence of fungal infections. So let's do some questions. One of the characteristics of fungal cells that ensure selective toxicity to antifungal agents is the presence of DAS, which is absent in human. Chitin, peptidoglycan, cholesterol, lipoic acid, nystatin. Remember we said the cell contains chitin, glucan, and mannan, and chitin is not present in humans, so you can use this to be able to target it using antifungals. Nystatin is a drug. Lipoic acid is found in the, it's not only found in the fungi. Cholesterol is found in human. Peptidoglycan layer is also going to be found in, but this is actually part of the cell membrane. One of the fungal cell characteristics is that they are vascular, motile, avascular, and undergo binary fission. So these guys, one of the fungal cell characteristics is that they are vascular. They don't have blood vessels. Motile, most of them, they're actually not going to move. Okay, they are immotile. They don't move. Of course, they can spread by hair, but they don't move because they don't have legs. They don't have blood vessels, so that's avascular. They undergo binary fission. We know this is also true. Okay, so one of the fungal cell characteristics is that they are undergo binary. Okay, this D is not true. It's false. They don't undergo binary fusion, but binary fission. Fusion is coming together. Fission is breaking. This is how questions come, and then you get to confuse them, right? The following antifungal agent is an example of an heterocyclic benzofuran. So heterocyclic benzofuran, we need to know glycofulvin, this guy that we said that it is going to disturb cell division, and it's only used against dermatophytes. The other term for pyrimidine analog is called, so the pyrimidine analogs, we call these are the antimetabolites. Okay. For example, what did we mention? We mentioned the flu cytosine. It's an antimetabolite. The following antifungals are correct paired example of triazoles. Remember we said that at least we should just know the imidazoles. Imidazoles we said BCKM, right? So anything where there is BCKM, B is C is cotrimazole, so this one is out. And then C is cotrimazole. Even here we have got cotrimazole and miconazole, so these ones are imidazoles. And then here we still have B-conazole, which is there. So these BCKM, these are what? Imidazoles. So the rest now will be the triazoles with 3 carbons. Fluconazole, itraconazole. What are the other examples? Posaconazole, voriconazole. Right? The group of antifungal agents that work by inhibiting glucan synthase, what do we call those? Those are echinocandins, right? Polyenes, these ones are going to bind to egosterol. And then you have got this heterocyclic benzofurans. This is where you have got glycio, the one that prevents the cell division, glyciofulvin. And then the antimetabolite you have got flu cytosine, which prevents nucleic acid synthesis. The azo and fungal agent work inhibiting the conversion of. How do azos work? Remember, it is a lanosterol to egosterol. Which one works by preventing the conversion of squalene to lanosterol? We call those as the allyl amines. The following antifungals are correct paired example of imidazoles. They contain two nitrogen atoms. The imidazoles, remember we said BCKM, so it is chlorotrimazole and ketoconazole. Okay, because here we have got fluconazole, which we know is a imidazole. Bifoconazole, of course, it is itraconazole. It is also an imidazole. An example of pyrimidine analog and fungal is, so the pyrimidine analog, we have got citarabine. Citarabine. So one we just need to know here. We said this pyrimidine analog, they are the antimetabolites. For example, flucytosine. Flucytosine is converted to 5-fluorouracil, and then it will be able to do its work of preventing the production of nucleic acid. The following antifungal agents are structurally classified as antibiotic-like. So the antibiotic-like, we have got nystatin, these guys that are inhibiting the cell. So nystatin is an antibiotic-like. The poly, the what's the name? The polyene antifungals. Okay, so that is it for this topic. Thank you so much for being present.